Welcome to this second of the Prather lectures in biology. This is a three-part lecture series. Yesterday we heard the first of these lectures in Sanders Theater in the evening on biodiversity. Today our subject will be the superorganism, and tomorrow here again at 4 o'clock, the third lecture on consilience. So I hope to see you at all of them. I'm very honored today to have the opportunity to introduce my longtime mentor, Edward O. Wilson. When I was a graduate student here at Harvard, Professor Wilson was fond of telling us that graduate students came in two varieties. At one extreme was a student who came in with a passion for a group of organisms, let's say ants, for one. The student wanted to learn everything possible about this group of organisms and built a thesis around that subject. At the other end of this spectrum was a student who came in with an idea or a hypothesis that he or she wanted to test. And their goal was to find the perfect group of organisms or the perfect study system to test that idea. Professor Wilson pointed out that both of these two approaches to studying the natural world were perfectly legitimate, and he celebrated both the empiricists on the one hand and the theoreticians on the other. He was also very influential. I came to Harvard with a lifetime interest in butterflies, and now I have a lifetime interest in butterflies and ants. At the same time, at the time, as I was a student, it never occurred to me to ask on which end of the spectrum Professor Wilson fell himself. But it's clear to me that, rather remarkably, this remarkable scientist falls on both. He's the world's leading authority on ants and on social insects in general, and yet he's never lost track of the big picture. And again, he's marshaled enormous amounts of information and data to test different ideas, different hypotheses about life. Unfortunately for us, while most of us labor away on papers, Professor Wilson prefers to write books. Just thinking about some of the titles makes my point. In 1967, The Theory of Island Biogeography with Robert MacArthur. In 1971, The Insect Societies, a book that launched careers for many, many people in social insects, including myself. In 1975, Sociobiology, The New Synthesis, heralded as perhaps the most influential book on animal behavior and behavioral ecology ever written. In 79, On Human Nature. In 84, Biophilia, with great medical significance. And I hear today he's winning an award for architecture because of biophilia. In 92, The Diversity of Life, heralding studies of biodiversity. And most recently, in 2009, with his longtime collaborator, Bert Haldubler, The Superorganism, The Beauty, Elegance, and Strangeness of Insect Societies. Professor Wilson's extraordinary gift to synthesize the many wonderful details of natural history into powerful theoretical frameworks has been the hallmark of his work. He has other traits, too, which I'd like to illustrate with a very quick story. Recently, I had a graduate student, Corey Moreau, who came to Harvard to study ants. She was definitely on one end of that spectrum. Every part of her body, I was told, was tattooed with different kinds of ants, beautiful ants. So about a month after Corey began work, so she came here to study ants, and we decided it would be fun to do a phylogeny of the ants, an evolutionary history of the ants. And we were thrilled when Corey got a small grant, about $40,000, to do an evolutionary history of the ants. About a month after she started work on this phylogeny, news came that a distinguished group of ant systematists, including representatives from the 3 major museums around the country, the Smithsonian, the American Museum, the California Academy, had received a $3 million grant from National Science Foundation, Tree of Life, to do essentially the same project, the phylogeny of the ants. This was a crushing disappointment, but it was still early in the research, and since training students was part of this whole idea, we contacted the people at the Tree of Life, and we said, how about we join forces? We'll give you all our ants and the sequences that we have so far, if you just let Corey work with you, and she can work on one of the some 20 subfamilies of ants, and we'll give you everything. I'm sorry to say that the Tree of Life team declined this offer. And to be fair, I think it was just too difficult for them to reformulate their plans to include an unanticipated first-year graduate student from another institution who wanted to work with them. Well, this is where Professor Wilson entered the picture. Corey and I met with him in his office to tell him the very sorry story of how our aspirations had been cut short by this series of events. And I still remember the puzzled expression on his face when he looked at us and said, "Well, I don't really see what the problem is. You did the right thing. You offered them everything, and they declined. I think you're entirely in your rights now to just go ahead and do the project." So Corey and I looked at each other at that moment and realized that this had never been in our worldview. we had backed down completely. We had never thought about David against Goliath. But we walked out of that office and we sequenced ants for 8 months straight, and so did everybody else in the lab. And eventually the paper made it to the cover of Science. So this story tells you several things about Professor Wilson. And for those of you who've read his recent novel, Anthill, you might recognize a few traits from the main character. He gives great advice, he's always fair in a fight, and he never gives up. So I'm very happy to welcome him here today.
Thank you. Thank you so much. I'll add to the amazing Corey Salk's story that she did the whole thing. She did it in 2 years.
8 months?
8? Well, I mean, there's 21 families of ants. But the team was going to spend years going. But to do it, this amazing second-year graduate student by that time came and said, "You know, I think I can do the whole thing myself." But she says, "I'll need a postdoc." So I thought through my head quickly about this, and I said, "Yeah, I can come up with the funds to get her a postdoc." She knew someone that really wanted to collaborate with her, and she did it. And so now it was the team, the establishment, as in the Human Genome Project, racing this graduate student. And she beat them. I mean, she was on the cover of Science, and they made it just in time to publish their phylogeny by the end of the year so that we can fairly say that independently, you know, they arrived at the same major phylogeny. And I'll tell you, was I relieved when they were identical? There was some disagreement about one little basal group. They'd come to the same conclusion. That's a remarkable story in Science, and yes, that's what I'm going to do, focus on ants tonight. So let me begin immediately by answering the most frequent question I'm asked, which is, what can I do about the ants in my kitchen? And my answer is, watch your step, be careful of little lives. They especially like honey, tuna, and pecan sandies cookies. So put down little bits of those and watch closely what happens, and you will see social behavior so strange that it is, and it might as well be on another planet. And that is the subject of my talk tonight. But first, a word of caution. There is nothing, let me repeat, nothing in the lives of ants and other superorganisms around us that we should aspire to emulate for our moral betterment. First of all of the ants you see are females. Males appear once a year, but they're pitiful creatures with wings, huge eyes and genitalia, and a small brain. They are virtually unable to feed themselves or do anything except be groomed and kept for a few weeks. They do no work, but have only one function in life, and that is to inseminate the virgin queens produced by other colonies. They are flying sperm missiles, and once launched, they are not allowed back home. They are born, they mate, they die. Now, I am an ardent feminist, but I consider that liberalism run amok. Second, many kinds of ants eat their dead. and they're injured. If you're injured and sick in an ant colony, you are programmed to leave the nest and not burden the society any further. Your sisters, that's what they all are, your sisters in there, are quite happy to see you leave. I suppose they're happy to see you leave. And ant experts call this the Republican strategy of health policy. Finally, ants are the most warlike of all animals. Colony against colony. The warriors, first in offense and defense, are all aging females. The sequence in the division of labor as they get older is such that when you get old, you get ready for war. So, here is a big difference between people and ants, where humans send their young men into battle, ants send their old ladies. Well, today, turning serious, if I can manage it, I'm going to use ants as paradigms of the origin of superorganisms, but first I need to speak about the process of evolution by natural selection. And at the very base in this is a distinction between what are emerging as the two laws of biology. Now, a law is a formulation that provides, when unpacked, without exception, detailed explanations of a broad system of phenomena. The first law of biology is that the entities and processes that define life are ultimately obedient to the laws of physics and chemistry. This principle is adamantine in biology. It is the keystone of molecular and cell biology. The second law of biology is that all the entities and processes of life and all of its diversity are the products of evolution by natural selection. That is, the increase of randomly generated genes that result in higher levels of reproduction. Some of you who have backgrounds in biology or genetics may think that you can come up with exceptions to that, but you would be wrong. These two laws, taken together, lead to a principle of evolution. The sharp distinction between the units of heredity and the targets of natural selection. The units are the genes, respectively, the base pair sequences and their codes, their location in the genome, and their interactions during the epigenesis of the phenotypes they ultimately prescribe. That then brings me to a second principle, namely that the targets of natural selection are the traits prescribed at every level of biological organization. Natural selection is, in a word, multi-level. These different levels of biological organization, shown in the next slide, are hierarchical. They've been reached one by one from the next one below only after geologically long periods of time. These are the major transitions of biology, major steps in evolution. Social insects are the steps that lead from organisms to superorganisms. The importance of the superorganisms, especially ants and other social insects, also comes, and I'll speak of this, what the step is in a moment, also comes from ecological dominance. In the Brazilian rainforest, for example, and probably many other parts of the world, they make up an astonishing 3/4 of the insect biomass. Although they only make up 2% roughly of the numbers of species, they make up 3/4 of the insect biomass, and over 1/4 of the entire animal biomass. They are almost everywhere. The principal turners of the soil are these social insects, especially the ants, predators of other small arthropods, and scavengers of dead animals and vegetation. For example, the ants in the Amazon forest, where the actual weights have been made, the counts have been made, ants outweigh all of the land vertebrates taken together, mammals, birds, reptiles, amphibians. All of those, they outweigh them 4 to 1. If you just go out and gather them all up and weigh them. In basic biology, the major transition from the organism to the superorganism is the easiest to study. I'm going to go back to those transitions, and has begun to provide important insights about how the big steps of evolution can occur. Now, a superorganism is a group of organisms so tightly united by altruistic division of labor between reproductives and non-reproductives that act in unison like one large organism, hence superorganism, that we can unblushingly refer to it as a superorganism. In approaching these complex systems, and especially their origin, I believe we have been, and this is a new development I just want to mention in passing, I believe that we, including myself, have been asking the wrong question for the past 50 years about altruism and the superorganism. We've been fixated on pedigree kinship. But there are now powerful mathematical analyses and evidence re-examined and developed here at Harvard, I might add, in progress, that kinship plays at most a superfluous role. The important question is in the origin of the superorganism is not the role of kinship, it is the rarity of the evolution of the origin of the superorganism. This is a complex issue in itself. It turns out more complex than we imagined. And by field and laboratory research and new mathematical modeling, we're beginning at last to understand what happened. It isn't too much to say that in this realm of evolutionary biology, we are, as I speak, in the beginning of a paradigm shift. In a nutshell, the rarity is due first to the necessity of a relatively rare pre-adaptation, that is, course of evolution in the solitary state up to a certain point, which is not in any way drawn by the future of the superorganism making appearance. That's not the way evolution works. It is an accidental approach in the course of adaptive radiations among many species, step by step, until finally the threshold leading to altruistic eusociality, the superorganism, is close by in the course of evolution. The change can occur then, put it over, by a single mutation, and the process can best be described, and this is part of the new approach, by extensions of classical population genetics, even of a diallelic system, with multi-level selection operating at different levels of biological organization, the group and the individual, acting on group and group members simultaneously. Group selection is merely selection acting on emergent phenotypic traits, which are unique to those that occur from the interaction of members of the group. That's the way to define it. And that way leads us out of the fever swamps of semantic difficulty in talking about group selection. But wait on this. There is hopefully forthcoming in a month or so in a leading journal a full exposition by Martin Novak, Corina Tornita, and myself, bringing, I hope, new excitement to evolutionary theory. Not all of the excitement will consist of cheering. Meanwhile, the beauty of ants, ants and the social insects generally, is, that is the intellectual beauty, the scientific beauty of them, is that we can clarify, and we have actually worked out the nature of the emergent traits. The systems are that simple, and the major transition from organism to superorganism is simple enough, an evolutionary step, although it has profound consequences, that it can be understood in the terms of mostly genetically tightly programmed behavior patterns. And we can do it. We can follow it precisely. And we also can use what I've called the pseudo-mutant method. When you make an of experimentation, you make do an experiment with an organism. You have to, of course, be invasive. You've got to take a cell out of a blastula, or you have to go in and inject with a higher titer of hormone, or deprive a little rhesus infant of its mother. You know, you do things like that. And that is very difficult to create controls for, and it is often technically very difficult to limit in a precise, repeatable way the change in the particular parameter needed in order to test a hypothesis about something that you are investigating. But in the case of looking at emergent traits and the superorganism as a unit, we can engage in the pseudo-mutant method, which we have done here at Harvard extensively. And that is, take a colony. You can start with one colony, and then you can do it again with another one, but actually you can do much of the work with one colony. And let's say it has several casts. Let's say it has 5 casts. You can take that colony, get it going in fairly uniform laboratory conditions, and then in some particular task, like caring for larvae, or in the case of the leafcutter ants, which I'll bring up shortly, getting leaves back to the colony, harvesting them and bringing them back. And in so doing, that latter task, you can measure how much energy, potential energy, in the leaves they bring back, fragments they bring back. And you can also get to that, the profit of that, the yield of that work. You can also get energetically the cost of it in manufacture of the ants, and also in the energies they're expending while they're doing the job. That's going to be that precise. But at any rate, how do you determine what the contribution is of different casts? That's easily done, because you get the ant, you get the colony spread out, either going in a trail or reaching inside, and you remove the caste that's, whose effect and efficiency in that caste you want to test. So now you have a mutant colony. I mean, it's identical to the colony you started with, but it lasts one caste. And now you study its performance, its responses, its performance, you measure its performance, and then at the end of the day, you put it back. It's almost, they put them back, and the next day you can do it again. And or you can move to another caste. And that's in fact what I've done. It's sort of the equivalent of now, you know, let us study the functioning and the importance, effectiveness of the human hand. So each morning, you know, you slice off a finger painlessly. The hand, you don't even notice it. And then you study, give the hand a test, and measure its performance and so on. And at dinner time, you put the finger back on. The next morning, the next one, you slice that off. That's basically what we're doing. I just thought for your amusement that you'd like to know that. That's that really. Now, having said that, I'll pass on for your delectation to some of the extreme products of group selection in the evolution of social behavior, and particularly of the superorganism. I want to start at the top with a highly complex ant system worked out in the laboratory and field by Berthold Obler and myself, the weaver ants of tropical forests in Africa and Asia. They live in the treetops. They are among, in tropical Africa and another species in tropical Asia. They form nests up there. They build them as pavilions with silk that they solicit from their own larvae, their growing sisters. These nests, as you can see here, are anchored onto twigs and leaves. They are immensely large structures in terms of the amount of silk needed to create them. They are almost invulnerable to predators and parasites because the ants are extremely aggressive. In World War II, the U.S. Marine Corps, and I suppose the Japanese too, came up against, particularly when snipers went up in the trees, came up against the tree ants. And Marines, more than one, I've talked to some veterans of this, they said, we were more afraid of those red tree ants than we were of the Japanese. They didn't want, they're ferocious, is what I'm trying to say. The colonies are up into the hundreds of thousands of workers. They dominate, each colony dominates the canopy of a tree or more than one tree. And they build their nest in a remarkable form of coordination in which they form chains of their own bodies as depicted here. And they, the thicker the rope that they build out of their own bodies with all the ants in pulling, or sheet, as in the top panel, where they're pulling two leaves together, the edges are together, they just increase the number of ants pulling until they're able to move those leaves, entire leaves or the edges of leaves, to curl them. They pull them together and hold them as living, living clasp, just hang there until other ants come out carrying a larva, as in the middle there, a grub-like larva from the nest. And these larvae are at the final stage of their development. They're capable of spinning silk, which would, in other members of this subfamily of ants, the Formicinae, go to making their protective cocoon. But these larvae of this species are programmed to do otherwise. They do not spin a cocoon for their own protection, but rather they wait until an ant, a sister ant, picks them up, as shown here, takes them out to where the weaving needs to be done, and then moving them back and forth, using intricate signals with the antennae to the larvae to release the silk. Something worked out by Bert Holdover with his ultra-fast photography. The larva then releases the silk and becomes a living silk shuttle. The ant moves it back and forth until it expels the silk it can create. And the larvae, therefore, in total, construct the nest with their silk, as guided by the ants themselves. Bert and I, working in the laboratory, proceeded to puzzle out the chemical communication used by these ants. We thought it probably would be very advanced because the ants, overall, the tree ants, the Ecophylla ants of the old world tropic, their behavior and their organization appears to be as complex as that of a honeybee. So we figured this would be a very good model species to work out chemical communication. In the late 50s and early 60s, I'd begun here working on pheromones of ants. And that was a time when the technological breakthrough occurred. Technology always drives most of science, as you know. This one, which made it possible to measure the minute amounts of substances used by the ants in communication. And that method is one standard today, but it was just coming into use in the late 50s among natural products chemists, and it's with mass spectrometry, gas-coupled mass spectrometry, gas chromatography. As you know, that's used to identify by the patterns of peaks, quantities of substances down to a millionth or even a billionth of a gram. Now, those were the amounts that ants carry in their little bodies, which may be somewhere anywhere from half a milligram to 10 milligrams. And they, turns out, we learned over a period of time, and particularly in those early days, I had a wonderful collaboration with natural products chemists. We were the first really to see what the ants were using and how much they were using. And we found out that these ants are living batteries of exocrine glands, as illustrated in this diagram of the body of a weaver ant, showing where the exocrine glands are. And I won't bore you by a catalog of them, but needless to say that in the end, in the 60s passed by, and we learned more and more, and other investigators as well, ants communicate with roughly 10 to 20 classes of pheromones, substances. These are typically relatively simple pheromones, substances, but they have certain consistent values. Early on, Bill Bossert, who is on the faculty here, and he was a young, beginning applied mathematician, a very gifted one, and I collaborated to build a physical model, a physical model primarily, of the evolution of pheromones in ants. And we found that when we made predictions about what the functions were of the various pheromones, such as alarm, attraction, trail following, agitation or excitement. Fire ants, for example, use 5 pheromones, usually with different functions, to work up their sister workers and get them moving to go out on a trail. There were 10 to 20 of these broad categories. And as time went on and the subject of pheromone chemistry evolved further, and I think the social insects always had a lead in it, at least initially, in terms of getting details and technique. We found that they could combine these pheromones and get, like words, and get a different message, or at least a compound message. There could be a sequence in the pheromone release, almost like a sentence, that it was quite remarkable. And we were, thus these ants, like the Ecophila ants that Bert and I set out to study, they can convey an enormous amount of information. I think that Bert Holdobler and I were able to establish, with the help of chemist colleagues, that this is the most complex pheromone system in the ants, and I suspect of all animals. In fact, we found that they use different sets of pheromones and mechanical movements and stimuli to pull sister workers out variously to a new nest site that a scout has discovered, to an enemy that needs to be combated, to a new food source. They use different signals and combination of signals to convey those messages. And here are some of the ways and some of the glands on the right that are involved in that highly sophisticated system of a code of chemical communication the ants use. More than one author, ourselves included, have noted the analogy between the pheromones of these ants and pheromones used by other animals with the hormones communicating among the cells and tissues within the organism. I think it's a pretty tight analogy. We now come to a species of ant that Bert Holdobler himself worked on the most. I didn't join in this part of it. In the Harvard laboratory while he was here, he kept colonies of the honeypot ants. These are ants found in the southwestern desert, which have a special caste. These are the creatures hanging from the nest ceiling in this drawing, which are fed large quantities of honeydew and nectar that the worker scouts and foraging workers bring into the nest. Honeydew is the sugary excrement that is produced by sap-sucking insects like aphids and scale insects and so on, and is the foundation of the tight and extremely efficient and remarkably complex symbioses between those two great groups of insects, which Nomi Pierce and her group have led in the study of in field and in the laboratory. Well, why are they doing this? These are living casks of nutrients, liquid nutrients, living. And these ants live in the desert, and during the dry season, when they cannot forage, they stay within the nest. In effect, they estivate. And these honey pot ants, we call them, the honeypot ants, the workers, hanging from the ceiling, regurgitate food upon demand, and they take the colony through the hard times. Oh yes, now we move to, I want to make sure I got the right one, yes, to the group that I devoted myself to during the time that approximately that Bert was working out the wonders of the honeypot ants. I was doing a deep analysis of division of labor and communication in the leaf-cutting ants. And here we have the rival of these leaf cutters, I'm sorry, of the weaver ants and the honeybees in complexity and sophistication of the social behavior. And what you see here, going from the top to 2 to 3 and then down to 4 and 5 and 6 at the bottom, is an assembly line. These are leafcutter ants. They are the ants which you can see every time, you can't avoid seeing them actually, every time you go into the New World tropics, from tropical Mexico, Central America to South America, that go out in enormous columns, sometimes as much as 100 or even 200 meters from their gigantic nest, and cut leaves and flowers and bring them back, as shown in the top panel, to the nest. They bring them in, not as food, as was thought. They can't digest raw vegetation, but rather as a substrate on which to raise symbiotic fungi. They then, the workers that bring them in are a certain size and age, and I showed that, energetically that's the most efficient size and so on. They bring them in and pass those larger fragments to smaller fragments, as depicted in number 2, which cut them up into fragments about 1 millimeter across, and pass them on, I said an assembly line, pass them on as depicted in 3 to ones that chew them up into a pulpy material and add digestive enzymes to them, and 4 and 5, still smaller casts, plant these globs of tissue, a substrate, processed substrate, into the fungus nest, or the fungus bed, which is a honeycomb-like, sponge-like, fluffy material that's white because of the mycelia of the fungi growing all through the walls. The ants then feed on those fungi. They also feed, to some extent, on plant sap. They pluck the smallest ants down there on the side, little dwarf gardener ants that are there in enormous numbers, and they tin the fungi. They pluck out weed fungi. They carry about and replant hyphae, strands of the symbiotic fungi. They do not allow any other species of fungus to grow there. There is a parasitic fungus, the dread enemy of the fungus-growing ants, that's constantly getting itself established and it has to be fought all the time. Some of these species of the leafcutter ants have masses of bacterial colonies over their bodies, at first on the sternum, and then they even grow around the whole body to give the ant a frosty look. Those are bacteria that are producing antifungal agents that the parasite succumbs to, but the symbiotic fungus does not. And on top of that, there are, to complete just how complicated all this gets, there's a yeast species that's a parasite of the bacterial beds or mats on the bodies of the ants. And a whole system fits beautifully together, and the ants then have become the dominant herbivores of the New World tropics. They consume more, and here's the life cycle, I won't go into the details of it. They consume more vegetation than all of the other, than any other herbivore group. In other words, one colony of leaf-cutting ants consumes an equal amount of vegetation as a cow. Incidentally, just parenthetically, I'm bringing out a book shortly with a Spanish scholar, Jose Gomez, on the lost treatises of a great South American naturalist who preceded Humboldt named Jose Celestino Mutis, M-U-T-I-S. He's a national hero in Colombia. He's also famous in Spain and in Spanish-speaking countries, but we have never heard of him because nobody's ever published on anything with this great man in English. He lived in the arrived in 1761 in Bogota and died in 1808. He started the first astronomical observatory, which is still there. He was a mineralogical expert. He was the physician to the viceroy of the new kingdom of Granada, the Spanish colony which extended from Venezuela to Ecuador. He worked with a grant from the Crown to do the first botanical survey with magnificent, beautiful paintings, which are on display in Bogota, and have been published, many of them. And at the urging of Carl Linnaeus back in Sweden, who had seen some amazing ants sent by Rolander from Dutch Guiana to Linnaeus earlier, and Linnaeus suggested that Mutis have a look at these ants. He didn't know what they were like. No one knew anything about them. No one had ever studied anything about ants anywhere, except two people, one of whom never published in France, and another one had published just a little bit on English ants. So he undertook the study of these ants, of all their complicated system, and of the army ants, the other dominant, most conspicuous group that run in armies of hundreds of thousands. And he worked out a large part of the life cycle, some of it wrong, but a lot of it right. And he wrote a treatise which he dutifully sent off to Linnaeus, and it was lost at sea. So after years, he wrote a second one. That reached the Royal Botanical Garden of Madrid and was misplaced. Has never been found. So and then it was forgotten. The whole thing was forgotten until about 50 years ago, the Spanish scholars going through these archival materials from Bogota came upon his letters and his diaries and realized that there was material in it that undoubtedly served as his research notes. This was sent to me, translated into English, and I was asked to identify the ants that was being described if I could. And I wrote back and I said, I'll identify the ants, but I'm going to do more than that. We will publish a book on together on Mutis's, we'll publish Mutis's lost treatises. And that is what we're doing within the next few months from Johns Hopkins Press. And it'll be the first introduction of this amazing man to English reading audience. But also, and the reason I bring it up, is that we will give credit where credit is due, even though it didn't reach the light of day until the 21st century, the early work of this kind. He didn't have these details. He was often wrong, as I said. And he was the first naturalist, scientific naturalist of the Western Hemisphere. I thought I'd just mention that in passing. And this is what consumed his time, a lot of his time. And he was overwhelmed, as are we today. Only recently has a Brazilian team used the method of pouring concrete into the colonies of these gigantic nests that you can see. The mounds rise up 3 or 4 meters above the ground of excavated earth. So they poured in concrete to fill up the chambers and galleries of the nest, and then meticulously excavated to reveal the structure of the nest. And here you see the one of such nests that they have excavated. We're talking here about a colony with one queen. She lives, we know, for 13 years. Kathy Horton and I at the laboratory here have kept 2 colonies with a queen, young queen, and they both died at 13. So I figure we're close to what the longevity is. Others figure it's somewhere overall, according to species, around 10, 12, 15 years. During that time, the mother queen sits at the bottom of the colony, of the nest, when she makes it through and can establish a colony. And only 1 out of 10 queens, 10,000 queens that get out and mate with those poor, useless, doomed males. Only 1 out of 10,000 actually makes it to actually start a colony. But if she succeeds, one of these gigantic colonies will contain, at any given time, from 3 to 5 million workers, all daughters of that single queen. And she will produce a total of upwards of 150 million daughters during her lifespan. And then, of course, many of which will be new queens, and then there will be a lot of males as well. So this is what evolution of the superorganisms has wrought, and this is what we are trying to puzzle out at each stage that led up to these wonders of the animal world. We're working out using fossils, using phylogenies of the kind aforementioned from our amazing graduate student, and many other lines of evidence to figure out when they came into existence and how they came into existence, and even why they came into existence. And one of the great evolutionary problems that we are facing, to circle back now to the ongoing new assault on theory to crack open the mystery of how it's done genetically and by what kind of selection pressures, we want to figure out why it took so long. And I'm going to close by just telling you that something about the history of the insects generally. Insects appear as early as the Devonian period, way back in the Paleozoic. By the time of the coal forest periods and on into the Permian, the insects of the world go through a massive adaptive radiation. It produces the first beetles, the first dragonflies, the first mayflies, a number of other groups that have survived to the present day, cockroaches. It produces amazing groups that are now extinct, like the Ephemeroptera, the Paleodictyoptera, dragonflies with wings 3 feet across. It produces all these amazing things, obviously filling the niches, every niche made available by these early forests, swamp forests of the Paleozoic. But it produced no social insects. Now we have the end of the Permian. 90% supposedly of all species go extinct through the great spasm that occurs, and then the recovery in the early Mesozoic, and still no social insects. No social insects until the middle or toward the end of the Jurassic period, in the midst of yet another great radiation, possibly associated with the origins and the beginning of spread of angiosperms, the flowering plants, as, for example, My colleague Brian Farrell, in his work on beetle evolution, has demonstrated that many of the beetle groups, like the dominant plant-eating beetles and weevils and so on, burst to become the most diverse animals on Earth, in part in correlation with the new opportunity presented by the flowering plants. And it was about then, or a little before it, that finally the first ants evolved, the first bees, the first wasp. And so it's this, and of course if we call ourselves eusocial, if you can stretch the definition to call us humans eusocial, it took forever to produce a human being. Maybe it should have taken a few billion years longer, but here we are. Forever to build up the conditions, the circumstances, the ecological pressures, the genetic mechanisms to flip over some old world primates into the human line. And why did it take so long? Well, we're just beginning to find partial answers to some of these mysteries, but I hope that I've been able to use the social insects to good advantage today to show why, first, they excite such interest among biologists and from, others as well. And to brag about Harvard's contributions. If I can't brag here, where can I brag? The superorganism concept was first introduced, and the word first used by William Morton Wheeler here at Harvard in 1911. And so I and my 10,000 trillion constituents, that's the approximate number of ants alive at any moment, weighing about as much as all of the 6.6 billion human beings put together, we thank you most heartily, and do not forget the honey and tuna bits. You want to have questions? Okay, good.
Just a quick announcement while some of you, if you need to go, can go. Tomorrow night we'll have conciliants here at 4 o'clock in the same auditorium. And now we'll have a question-answer period. There'll be people walking up and down with a microphone. So if you have questions, just signal to somebody with a microphone, and Professor Wilson will answer the questions on his own. You'll just handle the question.
Yeah, let's try to keep it to 10 minutes, because they're going to be getting restless and leaving now, and I want to get home too.
Okay, so just we have about 10 or 15 minutes for questions.
Okay, fine. Go ahead. Yeah. Nice and loud.
Thank you very much for your lecture. The other transitions between, these major ones that you picked out, the eukaryotes, our development have seen instability in the Earth as a whole. There was the development of oxygen that came along with the eukaryotes. Are there events like that associated with the social insects as they developed, as they became dominant? Did they induce instabilities in the ecosystems?
Oh, did the origin of the social insects introduce instabilities?
Yes.
Is that what you're saying? I know because, none that we have any obvious sign of, because these, they didn't just suddenly appear. It took tens of millions of years longer to come up with things like army ants and leaf cutters and so on. The first ants were rather humble little creatures. They might have had small colonies, the Mesozoic ants, and then gradually we saw toward the end of the Mesozoic a radiation into the number of basic types, and we don't know much about the colony level, colony complexity, but it wasn't until well into the Eocene, with about, say, at least 50 million years ago, between 50 million and 65 million years ago, that ants really took over. You know, we had the lucky advanced type. So by this time, you know, when you have tens of millions of years to co-evolve, plants, the insects, and so on, and the group that will become world dominant is coming in gradually. I think that you don't get the impact that we have made. We are, if I understand what you're getting at.
Or cyanobacteria.
That is a unique event that one species like this ever, you know, really took over and did the kind of damage that it's done globally. It can be done globally. I said, okay, yeah. Yeah.
You talked about ants and human beings, and in particular about how successful ants were with respect to other insects. But I'm wondering about other groups that have demonstrated a little bit of social behavior. For example, naked mole rats or spiders of the genus Stegodiphyus. Most spiders don't, and obviously where I've seen Stegodiphyus, they don't knock out all other spiders. So
spiders never made it. Yeah.
What do you think in general about,
I don't know the answer
about
to
the
that.
groups where social behavior doesn't give any
it's,
adaptive advice?
That's right. There are lots and lots of species that made it through the suite of pre-adaptations, which I think we now understand pretty well, which is building a nest that's defensible, that's within easy foraging range, or foraging range, I should say, of food, but defensible against enemies. And when you have that, then you have lots and lots of organisms have done that. You know, solitary wasp and beetles of many kinds and so on. You have the right preconditions, but they still don't make the leap over. Spiders never did it. Spiders have had since the Paleozoic. And we know there is a wide variety of social spiders living together, and that they benefit by living together and having these mass webs that they catch larger prey in. And yet they haven't divided labor. They haven't developed a superorganism. And we don't know why that's the case.
Dr. Wilson, I was just curious to see if you could speak a bit about some of the disadvantages of becoming a eusocial insect or evolving eusociality within your group. Why are there so few cases reported within all of biodiversity?
Why are there so few? Oh, that's just what I said. That's the mystery of mysteries. I think I know the answer, but the point is that that's the key thing we should be focusing on. Why it happens so seldom, and therefore what very special conditions are needed. I think it's certainly not close related to genetic relatedness, because the history of life on Earth is just full of haplodiploid species. Well, maybe that really didn't matter after all, so the sisters are closer related. It hasn't evolved from, very rarely came out of clonal species in which the species are genetically identical and so on. So I don't think that's, all the evidence is against us. That's what's important. What's important is those steps that lead up to, because this is the one thing all of them have in common that actually made the jump. Defensible nest, they guard. And then progressive provisioning. You raise your young inside the defended nest. The vast majority of cases, the young emerge, and mom just goes this way, and the young go the other way, and they disperse. But in a very small number of cases, they stay together. And we know it only takes one mutation that would silence the tendency to disperse, and that'll do it. But then the little group that has decided not to disperse, the little elementary eusocial group, has got to make it as a group against all of these other dispersing groups, and that only happens once in a great while. There's a lot of more to this story. If you'd like to pick it up on it, it's in the first issue of the 2009 Bioscience article. I have a full review of all of this and other reasons why, it's so rare and how to go about studying it and so on. What about it?
Hi there. So if a group of bees or ants can be considered a superorganism, could, in theory, then the single queen or males be considered themselves supergametes for those superorganisms in
Say
terms
that
of
again,
modeling,
please.
et cetera?
Wait a minute, I didn't quite hear the last part. What did you say again?
Could the queens or males, queens and males, I guess in this case, be considered supergametes in a sense for the gametes, as in the sexual cells of the superorganism?
Supergenders.
Yes, gametes, as in like sperm and eggs, but on a larger
Yeah.
scale.
Well, the right way to think about it is that, in fact, and we have thought about it. In fact, even Darwin, in The Origin of Species, solved to his satisfaction what he called the greatest obstacle to the theory of evolution by natural selection, and that's the existence of worker ants. That he couldn't figure out, how worker ants if they're sterile, could give rise to worker ant. Well, he figured it out that it was really the queen to the queen to the queen, and that the queen could be thought of as a plant that a gardener is cultivating. And the gardener is cultivating the plant according to the qualities and selecting among the plants according to the qualities of the vegetable it obtains. The vegetable it obtains is the analogy of the worker caste. So the queen that can produce a worker caste, and mark you, as far as caste, the differences occurring queen to worker. That's the principle. That's a fact I didn't mention. They are identical genetically. They may differ in other qualities, but they seem to be identical genetically. That's held steadily. So the queen is producing another queen by generating an extension of herself in all these workers running around, and that's the right way. That's the way to look at it, I think. Okay. Somebody else then?
Thank you for your talk, Dr. Wilson. That was wonderful. I wondered if you might talk about how your studies of the evolution of the superorganism among insect societies gives you any insight into the evolutionary processes that might have created another eusocial animal that provisions its young, which lives in central places. I'm speaking of humans, and how the evolution of our form of eusociality came about, and whether you think what types of analogies in the insect world you can use.
Yes. What can we learn from the study of origin of eusociality in other creatures, and particularly including not just what is produced, has been produced in evolution, but what are the steps leading up to it, and what can we deduce about that from the origin of humanity? Did humanity's advanced social condition originate in any particular way? For example, was it the sudden development of a productive language, with arbitrary symbols that allowed people to suddenly get together by some form of reasoning and so on? And that's the subject of the book I'm working on. It's called The Social Conquest of Earth. And it's, can we go from all these animals that we now understand better and better to the explanation of humans? And I think we need to do it because I think that we should keep in mind, this is possibly a provocative statement, but I should think we should keep in mind that the central questions of philosophy are, and they always have been, the ones inscribed on Gauguin's Tahitian masterpiece on the canvas, which is, where did we come from? Who are we, and where are we going? I think we need to readdress those questions straight on, and if we need to use science to do that. And I'll tell you what I think. I think that since the evolution of animals, eusociality has always involved group against group level selection, and that generally speaking, competition among established eusocial colonies of the same species is intense. up to and including warfare. It's general, that we should consider the possibility that that's what created us. Indeed, probably the most politically incorrect thing that Darwin ever said, and it's almost never quoted, it's in The Descent of Man. He said, perhaps that those qualities of humanity that we most cherish, of honor, of bravery, of high intelligence, and so on, could be the result of the clash between groups in which only those qualities succeed in prevailing over long periods of evolutionary time. That was Darwin, and I think that's possibly what we may find out. And if we find it out, it's going to be, at first, a little bit of an unpleasant truth. In fact, we haven't caught on to it already. And we are the evolutionary product of persistent conflict. So if that is the case, then perhaps the solution of so many of our problems would be in understanding ourselves and our origins first, and then deciding the best way to get around it.
Ed, can I suggest that maybe that's a good place to stop, and thank you all very much for coming.
Thank you. Okay.